and get into chapter 4. And you should remember something like this from last class. We were talking about some of our passive processes, and we started talking about diffusion. Diffusion is the movement of solutes from an area of high concentration or greater concentration to an area of lesser or low concentration. And that's what we're seeing here in this picture, right? Our molecules are highly concentrated in this area, so they want to move outwards, and that's exactly what they do until they reach equilibrium, in which our solutes are evenly distributed throughout the environment there, the beaker. So that's what we're talking about when we're talking about diffusion. So when we're talking about diffusion, there's two types of diffusion that we need to discuss. The first is simple diffusion. And this is going to be the discussion how molecules, again, because when we're talking about diffusion, it's the movement of a solute, all right? So how the molecules are going to pass through the plasma membrane. Literally, they're going in between those phospholipid molecules. So that means that these particular molecules, these solutes, have to be really small, and they also have to be nonpolar. They have to contain both of these qualities for simple diffusion. So we see that in oxygen and carbon dioxide, which are respiratory gases. We'll see that in ethanol, urea, which is a waste product made by cells, right? It's made up of nitrogen, hydrogen atoms. So those molecules are very small and they're nonpolar, so they can move right through those phospholipid molecules. So that means that we are not regulating the movement of those solutes via the plasma membrane. The plasma membrane does not do any type of regulation on that. So that means that really what is going to influence the movement of these molecules or these solutes is the concentration gradient. So if we have a concentration gradient that's in existence, that means we'll see a movement of things. But if there's no concentration gradient, which means we have the same amount of solutes on either side of that plasma membrane, then we're not going to see any movement because it's reached equilibrium. And you remember, two things will influence the rate of diffusion. One is the temperature. If you want to heat things up and have the process go faster, fine, we'll increase the temperature, molecules will get more excited, and they'll move quicker. The rate of diffusion will be quicker. Also, if we have a steeper concentration gradient or a bigger difference, meaning if you have 100 of one molecule outside the cell and then one of that molecule on the inside of the cell, the difference is 99. And so you'll have a pretty fast rate of diffusion. Now, mind you, if we had 100, all right, no, excuse me, if we had 55 molecules on the outside of the cell and 45 on the inside, then it's only a difference of 10, so the rate of diffusion won't be as much. So that goes back to this slide here. Okay, rate of diffusion, all right, depends on the steepness of the concentration gradient. That's going to be the difference between the two areas. And then our temperature. We increase the temperature, we'll get more movement. Okay, so here's that picture of simple diffusion in regards to our respiratory gases. So if you look here, all right, there's the plasma membrane. On the outside of our plasma membrane, in the interstitial fluid, that's the fluid that sits outside of the cells, we have a lot of oxygen. On the inside of the cell, in the cytosol, that's the intracellular fluid, we have a couple molecules of oxygen. But you'll notice there's more on the outside than there are on the inside. So oxygen goes from an area of higher concentration to lower concentration, and it moves right through the phospholipid membrane there. And then vice versa, you'll notice all these carbon dioxide molecules here on the inside of our cell and a couple on the outside. So again, carbon dioxide is a waste product of cell metabolism. And so you'll see that the carbon dioxide molecules are influenced to move outside the cell, down their concentration gradient, outside the cell, into the capillaries, and then back to the lungs where you can get rid of it that way. So that's simple diffusion. The other type of diffusion is called facilitated diffusion. And this is the type of diffusion where the movement of these solutes needs a little bit of help, requires some assistance. And guess who's going to help out? Yes, you guessed it. Our proteins, the plasma membrane proteins. All right. There's a reason why we have them, many reasons why we have them in the plasma membrane. And one of those is to help facilitate the movement of solutes across the plasma membrane. So the type of molecules, and you should know this, that are going to be influenced by facilitated diffusion are going to be small charged solutes or polar solutes. What's a charged molecule? An ion. So cations and anions are going to require facilitated diffusion. Sodium, potassium, calcium, chloride. All right, all of those are charged particles. Okay, so they'll need facilitated diffusion. There's two types. There's channel-mediated diffusion and then carrier-mediated diffusion. So let's talk about channel-mediated diffusion first. All right, so we have these protein channels that are embedded into the plasma membrane. And so there'll be like a tube that runs down the center of it, and it's filled with water. And so it allows for the movement of small ions. So let's use an example of a small ion, sodium, because we're going to talk a lot about sodium in chapter 12 and in chapter 10. Na plus. All right, that's sodium. Now, sodium is in high concentration outside the cell. It's in low concentration inside the cell. So it wants to move from the outside the cell towards the inside the cell. So it's going to use the channels to help with that. An important fact that you need to know is that these channels are specific for one type of ion. That was horrible. Let me get rid of that. All right, they're specific for one type of ion. That means if that channel is specific for sodium, potassium can't go through, calcium can't go through, chloride can't go through. All right, so channels are specific for one specific type of ion. And so that's why we name those channels after the ion. So for example, you'll hear me use terms later on in the course, voltage gated sodium channels. And so I'm telling you what passes through that channel, which is sodium. All right, so there's two types of channels. There are the leak channels, and they're like 7-Eleven, which means they're open 24/7, all the time. So they're always open. And then you've got a gated channel. And most of the time it's closed. And the only time that it's open is in response to some sort of stimulus. And when it does open, it's only open for a brief moment. So these gated channels are like the gate in your backyard. All right, if the gate's open, the dog can get out. If the gate's closed, the dog can't get out. So that's how it works. Same thing here with these ions. If the gate is closed on these channels, the ion's not crossing over the plasma membrane. So this type of diffusion, all right, these channels here play a huge role in the function of both nerve cells and muscle cells. And again, chapter 12 and 10, we'll get into that in much more detail, so stay tuned. So the other type of facilitated diffusion is going to be carrier-mediated diffusion. And so we'll deal again here with small polar molecules. But now in this case, instead of having a channel, they're going to use a carrier protein. And what will happen is, say it's for glucose. So say we have a channel that is designed to get glucose inside the cell. So the glucose is going to come floating along, and there's a receptor on this carrier protein. And so the glucose will bind on to that carrier protein, and that will cause the carrier protein to change shape. And that shape change allows that glucose molecule to go from the outside of the cell to the inside of the cell. And that's essentially how it does it. Same principles, everything's going to move down the concentration gradient. If that carrier protein is only going to move one substance, we refer to it as a uniporter. And so this comes into consideration clinically for folks like with diabetes and whatnot. And you'll notice that diabetics in their urine will have increased amounts of glucose or sugar. And normally, in normal functioning people that don't have diabetes, right, when you take in a meal that has a lot of carbohydrates, for example, right, so your blood sugar levels will increase. And so the cells then, when insulin gets released, insulin, yes, helps to move the glucose inside the cell because it does influence these carrier proteins, but it also causes the cells to make more carrier proteins to put into the plasma membrane so the cell can bring in more glucose, right? That's important. But unfortunately, as with a lot of things, eventually the cell reaches its max limit. And so it cannot transport any more glucose into the cell because the plasma membrane is full of these carrier proteins. So what happens? Well, glucose can't enter into the cell. It stays in the blood. It makes its way eventually into the kidneys. It gets filtered out of the blood in the kidneys and into the urine. And then that is one of the reasons why you will see increased amounts of glucose, all right, or sugar in the urine. So that, keep in mind, all right, when we're dealing with the number of channels and these carriers, you can reach a maximum rate of the amount of that substance that are being transported across the plasma membrane. All right, so this is what our carrier-mediated diffusion looks like, just like this. So here we have, all right, our glucose is outside the cell. All right, it comes in contact here with the carrier protein. And you can see how the actual protein itself changes shape. It looks like it's a funnel shape facing out of the cell. Then the glucose, all right, makes contact and it enters into the protein and it starts to change its shape, eventually looking like this, where that funnel is facing into the cell. That's what we're talking about, that it causes the carrier protein, all right, to change shape. Okay, so that was the first type of passive process for transport across the plasma membrane in our cells. So now we're going to talk about the other type. And the other passive process is osmosis. Now, as soon as I say osmosis, or as soon as you read osmosis somewhere, the first thought in your head should be water. That osmosis is the movement of water and not of solutes. So if you're answering a test question and one of the answer choices is osmosis, but yet nowhere in that test question does it ever mention the movement of water, do not pick osmosis. Okay, it has to do with the movement of water. And it has to do with the movement of water through what we call a selectively permeable membrane. Well, guess what? Your plasma membrane is a selectively permeable membrane. So the plasma membrane is going to allow water to move, and I'll show you this in a second, okay? But it's going to prevent the movement of most of the solutes. Now notice I said most, because what doesn't it regulate? It doesn't regulate the movement of small and non-polar solutes. They can pass right through the plasma membrane, okay? That's why we say most, because if they're small and polar or small and charged, right, they can't pass right through the membrane unless they have help. So keep that in mind. The membrane allows for the passage of water. I got a picture here that I want to show you right here. Let me zoom in. All right, here you can see the movement of water. There's two ways that water can move across the plasma membrane. It can move right through the plasma membrane because it's permeable, or it can move through these special channels called aquaporins that are designed for water. So water can move in one of two ways, through the aquaporins or right through the plasma membrane. That is the two ways that water can move into the plasma membrane. All right, so keep in mind that if there are equal amounts of water on either side of the plasma membrane, then there's not going to be any movement, just like we saw with diffusion. Once we reach equilibrium, we don't have movement going on anymore. All right, so remember that. Don't forget it. Okay, so I already mentioned this, two ways that water can cross the plasma membrane, either going right through the phospholipid bilayer or through the aquaporins. Okay, so that's for water. So now we're going to deal with solutes here in a moment, because remember what I said, osmosis is the movement of water, but not the movement of solutes. But the solutes, and I'll get into this in a moment, are going to influence that movement of water in a second. So there's two types of solutes, permeable solutes and non-permeable solutes. Obviously, permeable solutes, and we talked about that in diffusion, will pass right through the lipid bilayer. So they have to have these two qualities, small and nonpolar. Enough on that. I've already said it. You know it. We're good. The non-permeable can't get through the lipid bilayer. All right, that means they're charged polar or charged ions, okay, or they're just too big. They're just too big. So those are going to be our non-permeable solutes. All right, this slide has a lot on it, so I'm going to kind of take my time and go over it here with you. Okay, so when we're dealing with osmosis, we have to establish that water will move if there's a difference in the concentration gradient. So we understand that that concentration gradient is going to exist across the plasma membrane. Specifically, it's going to be the concentration gradient between the cytosol, the intracellular fluid, and the fluid outside the cell, which is the interstitial fluid. So for our purposes for osmosis, water can move across the plasma membrane, but the solutes can't. So because of that concentration gradient, all right, we'll see that there's a movement of water. Okay, so here's where we get involved with solutes and how they influence the movement of water. Okay, let me just read this statement off to you because this is a very important statement. Greater concentration of solutes contains a lower concentration of water. Let me repeat that. Greater concentration of solutes contains a lower concentration of water. So now, let me go back to what osmosis is. It's the movement of water across a semi-permeable membrane, and water is going to move from an area of high water concentration to an area of low water concentration. I'm going to repeat that. Osmosis is the movement of water from an area of high water concentration to an area of low water concentration across a semi-permeable membrane. What is that semi-permeable membrane for our cells? The plasma membrane. So we're basically describing the movement of water across the plasma membrane. So going back to that circled statement that I have here, greater concentration of solutes contains a lower concentration of water. So if water is going to move from an area of high water concentration to an area of low water concentration, can't I say this statement, that water moves from an area of high water concentration to an area of high solute concentration? Because look at that statement. Greater concentration of solutes contains lower concentration of water. So water goes from an area of high concentration to low concentration. At the same time, I can say, all right, water is going to move towards the area where there are more solutes because there's less water. I need you folks to understand that. All right, so keep that part in mind because I'm gonna elaborate on this in a moment. Okay, so when we talk about the movement of water through osmosis, it is again dependent on the concentration gradient between what's inside the cell, the cytosol, and what's outside the cell, which will be the solution. Because if we take that cell and drop it into something. So water is going to continue to move until it reaches that equilibrium. So the example here on this slide here, it says water moves from solution of 1% solutes, which means it has a higher water concentration, into solution containing 3% solutes, which is a lower water concentration. Take that in for a second. Read that to yourselves here while I take a sip of my water. Let me show you in a drawing. I love my drawings. Well, I don't really love them because the artistic quality is not that good. But it's helpful. It's helpful. Okay, so here, let me draw a picture for you and see if I can help. All right, so here, let's talk about, we have two compartments. Compartment A, and then compartment B. That line in the middle, right there, that's our semi-permeable membrane or our plasma membrane. All right. So in compartment A, we're going to use that same example that we saw on the previous page. I've got a solution that is made up of 1% solutes. And then in my other compartments, I have a solution that's made up of 3% solutes. All right, so if I have a solution that has 1% solutes, all right, that means the other 99% of that solution is made up of, is made up of H2O. And then in compartment B, since I have 3% solutes in that solution, that means subtract 3% from 100, that leaves me with 97% H2O. All right, so if you add those numbers together in each compartment, they should equal 100%. Agreed? 1 plus 99, 100. 3 plus 97, 100. Okay, so and this is what now you folks need to pay attention, which of those compartments, and you can type it into your chat screen, which of those compartments has more water? Compartment A or compartment B? Type in your answer now, please. I'm very confident that everybody here is pretty decent at math. So, all right, so let's see. I already know that you all got this correct, of course. Okay, so good. All right, so knowing what we know about osmosis, which compartment does water want to move into? Remember, osmosis is the movement of water across a semi-permeable membrane from an area of high concentration to an area of low concentration. So looking at those numbers, where does water want to go? Does it want to move from A into the B compartment, or does it want to go from the B compartment into the A compartment? So just type in where you think water is going to wind up. Is it going to wind up in the A compartment, or is it going to wind up in the B compartment? All right, let's take a look and see what you got. That's right. It's going to move from A into B. Very good. Okay. So knowing that now, good job, everybody. Knowing that, so we're going to see that water is going to move. Water is going to move this, come on, this way. So now look up here now at the solutes. 1% solutes, 3% solutes. So if water is going to move from compartment A into compartment B, water is going to go from an area where there's only 1% solutes to an area where there's 3% solutes. It's going to move to the area where there's more solutes. That's what I'm talking about here in this statement. Greater concentration of solids contains a lower concentration of water. So that means that water is going to move to the area where there's a greater concentration of solutes because there's less water there. I hope you understand that. That's an important concept. It usually takes me a while to explain it to folks, but I have a feeling you guys are bright. Okay, so that leads me into the next part of osmosis, which is osmotic pressure. So when we talk about osmotic pressure, we're talking about the pressure, all right, exerted by the movement of water across that semi-permeable membrane. So it's the pressure that water creates as it's moving across the plasma membrane. And that pressure is due to, find my pen, the difference in the solution concentration. Okay, so the solution concentration means we're talking about everything outside of the cell. That's what we mean. Okay, so if there's a difference in the concentration gradient between the inside and the outside of the cell, right? As that water is moving, it's going to create pressure. Just like if you turn the faucet on and water starts running out, you turn that faucet on more, it increases the water pressure, more water will come out. Okay, same thing here when we're talking about pressure, right? That is exerted through the movement of water. And so if we have a greater difference, right, between the two areas there, we'll have more pressure. Okay, so that steeper that gradient is, obviously more water is going to move because of osmosis, and that's going to increase the osmotic pressure. So since we're talking about pressure, we should talk about this term here, hydrostatic pressure. And basically, this is the pressure on the pipes inside your house. So when water is moving through the pipes in your house, it's going to put pressure on the walls of those pipes. So the definition here is pressure exerted by a fluid on the inside wall of its container. And so a lot of times we refer to our blood vessels and lymphatic vessels, the tubes of your body. Okay, so here's your capillary. And so as blood is moving in the capillary, all right, it pushes on the walls here of the capillary. All fluid does. And so that force or that pressure that blood is exerting on the walls of your capillary or vein or artery, we refer to that as the hydrostatic pressure. And that's just basically the push of that fluid or whatever to move out of that vessel or container or whatever. You'll get into more of that in 211. All right, so here, this is just showing you, all right, when we're talking about osmotic pressure, this is showing you, here we have our container with two sides to it. There's our semi-permeable membrane here. Okay, the red dots with the blue dots attached to them, those are the water molecules, and then these black dots, those are going to be the solutes. Now, if you look, there's only 4 solutes here on the right side, on side B, and there's 8 solutes here on side A. So knowing what we know about what water wants to do, it wants to move from an area of high water concentration to low water concentration. Or we can say water wants to move to the area with a greater solute concentration. So there's 8 on this side, 4 on this side, so it's going to want to move from B to A. Because there's higher concentration of solutes here, but also there's a lower concentration of water molecules. You can count all the water molecules here, 4, 5, 6, 7, 8, 9, 10, 11, 12. All right, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. All right, so we see that there's more water here, less water here. We're going to go from B to A. And that's what you see. So that is osmotic pressure. Now, final concept involving osmosis, and then we'll be done, I promise you. All right, so we have another term that I need you to learn, and it's called tonicity. So when water moves in and out of cells, all right, that loss or gain of water is going to change the cell volume. Meaning, if more water moves into the cell, it's going to increase the cell volume. If water leaves the cell, it's going to decrease the cell volume. All right, so, and obviously, if we're increasing the cell volume, we'll increase the osmotic pressure, because remember, the movement of water, all right, is going to exert pressure. So increase cell volume, increase osmotic pressure. Decrease cell volume, decrease osmotic pressure. So we came up with this term here called tonicity. And I'm going to read the definition here for you. It's the ability of a solution to change the volume or the pressure, which is we refer to as tone, of a cell by osmosis. So basically what we're going to do is we're going to take a cell, and we're going to use red blood cells as an example. We're going to take a red blood cell and we're going to drop it into a solution in a beaker. Okay. And we have 3 beakers in front of us. And those are 3 different types of solution. And so we're going to see what the solution does to that cell. It's either going to increase, all right, the cell volume and the osmotic pressure, or it's going to decrease the cell volume and the osmotic pressure, or it's not going to do anything at all. So we're going to talk about that right now. So here are our concentrations of our solutions. We have what's called isotonic, hypotonic, and hypertonic. So let me go through some examples to help you to understand this concept a little bit. So the first type is called an isotonic solution. So this means when we compare, all right, the solute concentration of the cytosol, that's the inside of the cell, and the solution that we just dropped this cell into, they're the same. The exact same. All right. That means, all right, if there's no difference or a greater concentration of solutes in one area or the other, water's not going to move, because that means that the concentration of water is the same. All right, so if the solute concentration is the same both inside the cell and outside the cell, because what did we say? Water always wants to go towards the area of greater solute concentration. Well, if it's the same, it's not going to go anywhere. So there is no movement of water. And so we refer to that as isotonic erythrocytes or red blood cells. So saline, all right, normal saline, all right, which is basically salt water. And we use it quite often in IV drips for folks that might have experienced a loss of blood, for example. All right, if their blood volume decreases, they've been injured and they've been bleeding for a bit, and we want to increase the blood volume so their blood pressure doesn't drop down too much. We'll just give them some normal saline, and that increases their blood volume, which increases their blood pressure. So that's an isotonic solution. All right, so the next type of solution is a hypotonic solution, which means the solution that we're going to drop our cell into has a lower concentration of solutes. That means it has a higher concentration of water compared to the cytosol. So there's more water outside the cell than inside the cell. Also, we can say there's more solutes inside the cell than outside the cell. So water wants to obviously move down its concentration gradient. So it's going to go from an area of high water concentration to an area of low water concentration. Our higher water concentration is outside the cell. Our lower water concentration is inside the cell because there's more solutes inside the cell. So water goes from outside the cell, inside the cell. It increases the cell volume and it increases the pressure on the inside of the cell. And if too much water moves, we can pop our red blood cell. And when we rupture the red blood cell, all right, or the cell, we call it lysis. And we give it a special name when it's red blood cells, we call it hemolysis. All right, the last type of solution is the hypertonic solution. All right, so now we're taking our red blood cell and we're going to drop it into a solution that has a higher concentration of solutes than the cytosol, which means it has a lower water concentration than the cytosol. So that means, again, water is going to move down its concentration gradient. It's going to move from the inside of the cell towards the outside because there's less water outside, but yet it's a higher concentration of our solutes. So when that happens, water leaves the cell, it decreases the volume of the cell, and it decreases the pressure of the cell. And so the cell shrinks, and we call that crenation. Be familiar with this. You're going to get probably 1 to 2 questions on your test on this, and it's just good to know. All right, you don't want to hang the wrong type of saline, all right, solution on a patient for this reason. All right, normal saline, okay, but God forbid you put a saline solution on somebody that's 3%, all right, sodium chloride, that could be an issue. Okay, you can cause damage to those cells. So that's what we're seeing here in these pictures here. All right, here's our isotonic solution. We have the same number of solutes inside the cell compared to outside the cell. So that means the water concentration is the same inside the cell, outside the cell. No net movement of water. Nothing happens. Boring. All right, our hypotonic solution. Now we've dropped our cell into a solution that has less solutes outside the cell, meaning there's more solutes inside the cell. So there's more water outside the cell because of that, and less water inside the cell. So water goes from an area of high water concentration to an area of low water concentration, where there's a greater solute concentration inside the cell. Water goes into the cell, the cell swells up, and it could potentially burst, causing hemolysis. Then finally, we take our red blood cell and drop it into a hypertonic solution. All right, that means the solution has way more solutes than outside the cell than inside the cell. So water is going to leave because it wants to go towards the higher solute concentration because that's where there's a lower water concentration. So it leaves the cell shrinks, and we undergo crenation. All right, so what occurs to the tonicity of a cell when placed in a hypertonic solution? We just talked about it. Water leaves the cell. It decreases the volume and the pressure inside the cell. And so that will result in crenation. All right, that's the passive processes. Now let's get into the active processes. A lot, I know there's a lot. It's all good. You folks are doing great. Okay, so when we talk about the active processes, all right, keep in mind, all right, these active processes involve two things: active transport and vesicular transport. And vesicular transport is in reference to those little vesicles, all right, those little bubbles that we saw inside the cell. So all type of active transport requires energy. We need ATP for this to work. Passive does not require energy. Active transport requires the movement of a solute against or up its concentration gradient. Passive transport, all right, that was movement of a solute down its concentration gradient. So obviously we're going to use active transport to try to maintain or create these concentration gradients, both inside the cell and in the interstitial fluid. All right, so let's talk about the first type of active transport, and that's going to be the ion pumps. All right, so like the name implies, it's a pump, and if it's an ion, it's going to be moving ions, all right, across the plasma membrane. And so we have these specialized transport proteins, again, proteins come to the rescue, that are going to move ions in or out or in and out of a cell. And so what the nice thing about these types of transport proteins is that they help to maintain the concentration of certain ions on the inside of the cells, because it's important. All right, we might not want to have a certain type of ion on the inside of the cell. For example, all right, our red blood cells have these calcium ion pumps, and their job is to pump calcium out of the red blood cell, because as much as I love calcium, when I need a muscle contraction, all right, or for the nerve to function, I don't want calcium accumulating on the inside of my red blood cells, because when that happens, the red blood cell, the plasma membrane, becomes more rigid and stiff. And you might not know this, but red blood cells have to squeeze in through really, really small spaces sometimes. Those capillaries can only accommodate one red blood cell at a time for the most part. So, and in some cases, a red blood cell has to squeeze out somewhere. And so we want the plasma membrane to be as fluid and as flexible as possible. So calcium makes the cell membrane more rigid, so we got to get rid of it. So we have these calcium pumps in the plasma membrane that pumps out calcium and gets rid of it so those plasma membranes don't become stiff. The other type of ion pump is the famous sodium-potassium ion pump. We'll talk more about this when we get to chapter 12 and 10. All right, but basically, the sodium-potassium ion pump is going to utilize the movement of both potassium and sodium in and out of the cells. So what will happen is, okay, this pump is going to move sodium ions from the inside of the cell to the outside of the cell. As you can see here in the picture, you have 3 ions here, right? They enter into the pump, and then as the pump is doing its thing, it moves those sodium ions out of the cell. Right. Meanwhile, all right, this pump is going to move potassium ions to the inside of the cell. So once sodium leaves, potassium comes on into the pump and enters into the cell. I'll get into more of the details on that. But basically, this utilizes ATP. The ATP molecule, all right, will attach itself onto the ATP binding site of the pump. And when it does that, it causes the pump to change shape, right? And that allows the sodium to leave the cell. And then as a result of the shape change of the pump, the ATP molecule loses one of its 3 phosphates and it turns into ADP. And the phosphate stays, right? And then once the phosphate molecule pops off, the pump goes back to its original configuration, right? But prior to that phosphate popping off, the potassium enters in. And once that phosphate pops off, it goes back to its original shape, and then the potassium enters into the cell. We'll get into more details about that. All right, but again, this pump is going to move, all right, those ions, potassium and sodium, against their concentration gradient. So there's more sodium outside the cell than inside the cell, but yet this pump pushes sodium, all right, from the inside of the cell to the outside of the cell. So it moves against its concentration gradient. So for every single ATP molecule that this pump utilizes, it's responsible for moving 2 potassium ions into the cell and 3 sodium ions out of the cell. Every time. This is happening all the time as we're sitting here. It's going on nonstop. All right, so that the ion pumps is a form of primary active transport. Secondary active transport, right, is a little bit different here. Again, it's going to move substances against or up their concentration gradient. But now it's a little bit clever, because with secondary active transport, all right, these mechanisms are going to utilize the movement of 2 substances. But one of those substances is going to be moving up its concentration gradient. While it's doing that, it's going to generate kinetic energy. Kinetic energy is going to be that energy from the movement of those molecules. And so that kinetic energy is enough to move a second substance down its concentration gradient. Clever. Okay, so we have the movement of 2 substances. One is going to move up its concentration gradient, while the second one is going to piggyback off of the energy that it created through the movement of those molecules, and it's going to go down its concentration gradient. So there's 2 types: symport, which means the same. All right, that's when both of those substances move in the same direction. But understand that one is going up its concentration gradient while the other one's going down. Antiport, all right, those molecules are going in the opposite direction. But same rule applies. One of those molecules, all right, or substances is going against its concentration gradient while the other one goes down its concentration gradient. So now this leads me to the other part of the active processes for plasma membrane transport, and that is going to be vesicular transport. Remember back in chapter, well, actually it was the beginning of this chapter, and we were talking about the Golgi apparatus. The Golgi apparatus is going to take these modified proteins and package them up and send them to a destination, either in the plasma membrane, outside of the cell, or eventually they'll fuse with the lysosome and become the digestive enzymes of that lysosome. All right, well, that's what we're going to talk about now. So this process that you see here on your screen is exocytosis. This is when a secretory vesicle on the inside of the cell is going to merge and fuse with the plasma membrane, and then whatever's in that secretory vesicle is either going to be shot out of the cell or it's going to be incorporated into the plasma membrane. That's exocytosis. So that's where we're taking something that was in the cell and we're getting rid of it. And so this process of exocytosis is important when we get into neurotransmitter function. So keep that in mind. Exocytosis, exo is going to be exiting. Something was made in the cell and it's getting expelled out of the cell. So the other type of active process that we're going to spend a little bit more time on is called endocytosis. This is when there's something outside of the cell, something in the external environment that the cell really wants. And so it's going to take it and bring it inside. And usually it's going to be large substances. All right, large substances. And this process of endocytosis is very similar to exocytosis, except we're just moving in the opposite direction. So we'll see this in cellular digestion, all right? We'll also see this during that process of our plasma membrane for the fusion of the exocytosis. We might have, there's parts of the plasma membrane that got expelled, so we want to retrieve those, all right? And then also when we're trying to incorporate certain proteins into the plasma membrane, right, to affect some of the cellular processes that are going to be going on for that cell, whether it's cell signaling or if it is something along the lines for protein synthesis. Okay, so there's three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. You've heard me mention phagocytosis because when I was telling you about the cells in lab class, the white blood cells, the macrophage, that's the Pac-Man cell, that's how it manages to get rid of any pathogens or whatnot, or foreign proteins. It utilizes the process of phagocytosis. We nickname it cellular eating, and this is basically when the cell is going to come across a large particle, all right, and it's going to basically absorb it into the cell. And it starts off by forming these little arms or appendages, all right, which we call pseudopods. And they surround the particle and then eventually encapsulate it, and then basically it makes its own kind of vesicle, this membrane type of sac. And then it pulls it into the inside of the cell. And then that sac is going to fuse with the lysosome, and that lysosome will spill its digestive enzymes into that sac and pretty much break down and digest whatever was in there. So we see this quite a lot in our white blood cells. And that's what you're seeing here in our picture. Okay, so here's our cell, and it really, really wants, all right, that molecule that's outside, I shouldn't say molecule, that particle that's outside the cell. And so it shoots out these pseudopods, and they eventually surround the whole thing, and it pulls it in. All right, and then this vesicle will eventually fuse with the lysosome, and that lysosome will digest all that. That's phagocytosis. All right, pinocytosis, this is the cell drinking. So that means we're going to be dealing with liquid, fluid, all right? So you could have these small droplets in the interstitial fluid, all right, that have these dissolved solutes, a whole bunch of different types. It could be dissolved molecules, ions or whatnot. So you can see this on a much more, I don't want to say larger scale, but you can have several of these going on at the same time. All right, so we'll see this quite often with most of the cells in our body, but the problem is this process is relatively non-specific. So that means whatever that vesicle, whatever is in that vesicle is what that cell is getting, right? So meaning, in this picture here, say the cell only wants this thing. Okay, unfortunately, it's going to get all of this stuff. There you go. Yeah, all right, yeah. That's a good point. I like that. I think of fag like getting fat and eating, and pina like a pina colada. That's a really good way to remember that. I'm going to have to remember that. Pina colada. Thank you. So that's what we're seeing here in this picture with our pinocytosis. This should not say phagocytosis. That should say pinocytosis. Okay. So anyways, it is non-specific, but it's just going to take in all of these droplets here of whatever this stuff is, and then it makes these vesicles, and the same thing will happen. We can digest those vesicles with the lysosome, right, and then internalize those contents there. The last type, the receptor-mediated endocytosis. This one's interesting. We're going to see more of this when we get into the metabolism of fats. All right, you're going to hear of lipoproteins, these concepts that you've probably, when you've gone to your doctor and if they've ever drawn blood on you and they break down your cholesterol, you'll see things that say like HDLs and LDLs. You hear things like good cholesterol, bad cholesterol. So we're going to see, all right, with receptor mediated endocytosis, that's going to involve, all right, the example down here is the LDLs here. But basically what happens is on our plasma membrane, we have these receptors. And those receptors are specific, all right, for a certain type of molecule that's going to be in the interstitial fluid, all right? So maybe something in the blood leaves the blood, it moves into the interstitial fluid, and then it's going to come close to the cell and bind on to the receptor there. All right, so think of these receptors almost like just trying to grab onto something specific, all right, that's floating by, and then once it does, it's going to pull it to the inside of the cell. And so this process is great because it allows, all right, the cell to obtain large quantities of whatever that substance is. And that substance is going to be specific, otherwise it won't bind onto the receptor. So we see that when we talk about cholesterol metabolism. So what will happen is, all right, say the cell it wants cholesterol. Well, that cholesterol is going to bind onto this molecule, all right? It's called the low density lipoprotein, all right? That's our protein molecule that has, you know, a lipid group on it. And so while it's floating through the blood, all right, it'll exit out of the blood, say like in the liver, and you have these special cells, the hepatocytes in the liver will have these receptors specific for these LDLs or these low density lipoproteins. And so they'll bond with the receptors. And when that happens, right now we've captured, all right, the low density lipoprotein with its cholesterol a group attached to it. And then the cell will actually suck the whole thing inside. I'll show you a picture here. Okay, so, all right, so all these little things, well, the blue is going to work out better. All right, the blue is, those are our LDLs, our low density lipids. So these receptors are special for that. All right, so the low density lipids will attach onto these receptors, and when we get enough of them, then the cell plasma membrane will fold in on itself. And it makes these special vesicles, all right, and we call them special because on the outside of the vesicles, we label. That's all this is. This molecule is called clathrin, and it basically is labeling, all right, this vesicle as, hey, we have low density lipoproteins in here. And so this needs to undergo the lipoprotein metabolism. All right, so these guys get their own special labeling, and that's that clathrin coating. All right, that's it for the plasma membrane. You probably don't remember, but from last class, we were just talking about the three parts of the cell. All right, the three parts of the cell are the plasma membrane, the cytoplasm, and the nucleus. Well, we covered the cytoplasm already, and we just got done with the plasma membrane. So now let's finish up with the nucleus. Okay, so if you're looking at a cell in your microscope, the nucleus is hard to miss. It's the largest structure inside the cell, often referred to as the control center. All right, pretty much every single cell will have a nucleus. There's a couple exceptions. Some cells could have many, like skeletal muscle can have many nuclei per cell. Cardiac cells have 1 to 2, right? But for the most part, most cells will have 1. And then there's an exception on the other way, meaning that some cells won't have any nuclei. So a mature red blood cell will not have a nuclei in it, okay? So for the most part, if you remember when we were talking about the tissues, the nuclei will pretty much mirror the shape of the cell. So when we were talking about, for example, our squamous epithelium, it was that flat cell, and the nucleus was flat. All right, and then when we had our columnar epithelium, the cell, nice, tall, long cell or high cell, and the nucleus looked like an oval egg. All right, so for the most part, it will mirror the shape of the cell. But what's really important is, guess what? Guess what lives here? DNA, genetic blueprints. Basically, your DNA is the instruction manual to your body. It tells cells everything about you, how to make certain proteins. It's the instruction manual for everything. Okay, and that all lives in the nucleus. Well, our nucleus has its own covering, much like how the cell has the plasma membrane. Well, the nucleus has the nuclear envelope, and it's similar to the plasma membrane in which it's made up of that phospholipid bilayer or a double phospholipid membrane that goes around the nucleus. Well, our cell has cytoplasm, okay? Well, the nucleus has nucleoplasm. And so that's going to be on the inside. And so it's important because things are going to be floating around in there. And there's a lot of stuff. And again, we're not going to go into all the details, but I'm going to talk about some of it. But we're going to have stuff that's inside the nucleus that needs to get out. So we essentially have these holes in the nuclear envelope. We call those nuclear pores. And basically, all right, these are just passageways that allows for the movement of large molecules in and out of the nucleus. Okay, because sometimes you remember how we talked about the plasma membrane, how there's things that are permeable to it and things that are non-permeable. All right, well, similar to the nuclear envelope, all right, the nuclear pores are going to allow the movement, all right, of those large molecules in and out of the cell, like channels would or aquaporins would, but small and non-polar substances can pass through easily, okay? So if we look further inside the nucleus, we'll see another ball in the nucleus, and we call that the nucleolus. So it's like a ball within a ball, all right? And the nucleolus is where, and I mentioned this before, where we are going to make small and large ribosome subunits. And those small and large ribosome subunits are going to be used to make proteins. So they're packaged up in the, they're made in the nucleus, they're made in the nucleolus, and then they'll be exported out of there and get packaged up and built to make proteins out in the cytoplasm. All right, so here you can see our picture of the nucleus here. So here's your nucleus with the nuclear envelope, and you can see you have the nuclear pores, and that's going to allow for the movement of substances. Then, if you look closer, you can see, all right, our chromatin here. All right, that's our DNA. It's just all coiled up right now. And then you can see a smaller ball on the inside. That's the nucleolus. And our ribosomes are made in here. And they'll eventually wind up outside the nucleus. So let me quickly mention DNA. Okay. DNA is our genetic blueprint. All right, it is composed of what we call repeating monomers, just like how we saw repeating monomers in glycogen was just a bunch of glucose just strung together. All right, the repeating monomers in DNA are what we call nucleotides. And nucleotides, all right, are made up of a sugar molecule, a phosphate molecule, and then one of our four what we call nitrogenous bases. All right, so I think I got a picture in here. I'll show you here in a second. So for our DNA, those nucleotides will be attached to one another. And that configuration creates this structure, all right, that looks like a spiral ladder. And so you know how a ladder has the struts, which are the side pieces, and then the rungs are what you climb up on. So when we're talking about the struts, the side of our ladder, that is going to be made up of, all right, that sugar molecule and the phosphate molecules. And then the rungs are going to be any one of these four, all right, nitrogenous bases bonded to another nitrogenous base by a hydrogen bond. Okay, so notice here, when we talk about the DNA, all right, we'll see that this houses most of the genetic material of the cell. Right. Not all of it is going to be found in the nucleus. Some of it will be found in the mitochondria. But you should know, all right, when we're talking about humans, right, we have 46 chromosomes, or separate double-stranded DNA molecules, chromosomes. 46, you should know that number. 46, 46, 46. So what we're going to do is we're going to take that ladder and we're going to twist it around. And then eventually, if you think about it, our instruction manual, I mean, it's down to the slightest detail: the color of your hair, how tall you're going to be, what color your skin is going to be, your eyes, how well you make certain proteins. That instruction manual is big, it's thick. So your cells have managed to find a way to package up this instruction manual and pack it into one of the smallest structures of your body. And then we're going to stick it into an even smaller structure on the inside of that structure, in the nucleus. And so what we're going to do is we're going to wind, all right, this DNA around these proteins called histones. And then what will happen is these histones then will start to coil up into chromatin and then eventually form our chromosomes. I'll show you a picture here in case you're not following me. All right, so when the cell is not dividing, all right, we'll find your DNA in the shape of what we call a chromatin. When it is dividing, right, the DNA chromatin becomes so tightly coiled, all right, that it'll look almost like an, I'll show you later on, like an X. We call those chromosomes. So here's our picture. Let's get down to the small portion. Here's our ladder. We've just twisted it around. So here are the struts here, and then here are the rungs. All right, so the backbone here, all right, that's made up of that sugar group and the phosphate group. And then our nucleotides, or nitrogenous bases, excuse me, all right, they're going to make up the rungs here. And we call them complementary base pairing. Again, this is not a genetics course, so I don't expect you to know all this, all right? But this complementary base pairing, we're talking about DNA, is that certain nitrogenous bases will only bond to another specific type of nitrogenous base, all right? So adenine and thymine, all right, they'll bond with each other, okay? And cytosine and guanine will bond with each other. And that bond is through hydrogen bonds. All right, so this specific type of pairing is going to give you diversity in your genetic makeup. So now we've got to take this big ladder and we twist it up, and then we've got to package it up. And so what we do is we twist it up and wrap it around these proteins here. And then we start to create these coils. And these coils are now called the chromatin. And then that's in the non-dividing cell. When it's time to divide, then we're going to take that coiled chromatin and we're going to package it into a chromosome. That's later on, not in this chapter and not in 210, but you'll get to learn about that in 211. Okay, this is all I want you to know about that. All right, I'm not going to worry about that. Last slide here. Let's quickly just talk about cell division. Okay, real simple. All right, cell division is when one cell splits in half and gives you two cells. Okay, so one cell divides, you get two cells. And obviously, we're going to see that during growth periods, like when you're growing up as a little kid. All right, so for development, if you're getting rid of some old cells, all right, or if there are some damaged cells and we need to repair the tissue, that's when we'll see cell division. So there's two types of cell division, okay? Mitosis and meiosis. Let's start with meiosis, the second one there on the bottom. Meiosis, all right, is when you get cell division between sex cells. That's easy. Sperm and the egg cells, which are called oocytes. So let's be specific, secondary oocytes. So when sperm and secondary oocytes divide, that is meiosis. Mitosis is cell division for all the other cells in your body. That's how I remember it. Meiosis, sex cells. The other type of cell division is all the other cells in your body. All right, those cells are called somatic cells. And that cell division is referred to as mitosis. All right, so what is the function of the nuclear pores? Again, it's to allow the passage of particles in and out of the nucleus. The end. Okay. If you have any questions, hold those questions, all right, for lab. Let's take a break.